Page 35, Heredity and Genetics. Recap, Section A. Heredity is the passing of characteristics or traits from one generation to the next. Genetics is the scientific study of heredity. Chromosomes are found in the nucleus of a cell and are made of tightly wound strands of DNA. During mitosis and meiosis, chromosomes may be seen with a microscope as rod-shaped structures. Deoxyribonucleic acid, DNA, is a molecule that contains genetic information that is passed on from parent to offspring. The DNA molecule is shaped like a double helix. The model shown here was proposed by the scientists James Watson and Francis Crick. The molecule is made of sugars, phosphates, and pairs of nitrogenous bases, such as adenine and thymine, cytosine and guanine, arranged in a specific way to form a chemical code. During mitosis, a copy of the genetic code is made when each chromosome is duplicated. In this way, each new cell made during cell division contains the same genetic code as the parent cell. A change in the sequence of nitrogenous bases in the DNA that codes for a trait is called a mutation. Mutations may be helpful, harmful, or neutral to the organism. Mutations are passed to daughter cells during mitosis and meiosis. A change in the DNA during meiosis can result in a changed trait called a variation. Part B. Traits have various expressions in the members of a species. Variations include colors of hair, length of fingers, shapes of seed pods, and the heights of a plant. A gene occupies a specific location on a chromosome. It contains the code to produce a particular form of trait, also known as an allele. Many inherited traits have at least two different variations. An allele is a variation of a gene. One allele may mask or hide another variation of the same trait. We say that this trait is dominant over the other. The variation which is hidden is said to be recessive. Part C. In preparation for sexual reproduction, sex cells, egg and sperm, are formed during the process of meiosis. Sex cells carry only half of the genetic code needed to form a new individual. When sperm from the father joins an egg from the mother, a new organism develops with a full set of chromosomes in its cells. The unique combination of genes in the offspring determines the variation of the trait it will have. Genotype refers to the genetic makeup of the organism for a particular trait, that is, the specific forms of the genes or alleles which have been inherited from the parents. Letters are often used to represent the genes in a pair to understand the model the inheritance of traits. A capital letter represents a dominant allele. A lowercase letter represents a recessive allele. For example, a gene for flower color may have an allele that produces red flowers, which is dominant, capital R, over the allele that produces white flowers, little r. For organisms to have pairs of chromosomes, an organism which is hybrid or heterozygous for a particular trait has one dominant form of the gene and one recessive form of that gene. For the flower color example, the genotype of a hybrid red flower is capital R, lowercase r. An organism that has two genes, which are similar for a trait, is called a purebred or homozygous. The genotype for a purebred red flower is capital R, capital R. A purebred white flower is lowercase r, lowercase r. The visible trait, or the observable effect of the combination of genes, is referred to as phenotype. In the previous example, the phenotype of a flower may be red or white. A Punnett square is a model used to show the possible combinations of alleles from a single trait cross. It is used to predict the probability of the genotypes and phenotypes of offspring produced from a cross between two parents. You can see an example of a Punnett square below. Rose color inheritance can be complex because quite a few colors and genes are involved. However, the gene for pink color is dominant over the color white. The following example shows a hybrid cross of roses with pink flowers. The capital P represents a pink flower, the dominant trait. Lowercase p represents a white flower, the recessive trait. Plants with the genotypes capital P, and capital P, lowercase p will have pink flowers. Their phenotype is pink flowers. The Punnett square shows the probability of the offspring from this cross having pink flowers is 75%, or 3 out of 4. Plants with the genotype lowercase p will have white flowers. Their phenotype is white flowers. The probability of the offspring having white flowers is 25%, or 1 out of 4. The phenotypic ratio for the cross shown above is 3 pink to 1 white. The genotypic ratio is 1 capital P to 2 capital P lowercase p to 1 lowercase p. Section D. Some traits are not inherited and are referred to as acquired traits. Acquired traits are influenced by the environment. For instance, if a person learns to play a musical instrument, this ability will not be inherited by the person's children. No genes are changed by practicing the piano, so the acquired ability will not be passed to future generations. Other examples of acquired traits include food preferences, language spoken, music enjoyed, length of hair, and injuries. Section C. Some traits are the results of the interaction of heredity and the environment as illustrated by the following examples. 1. If a person inherits genes for tallness but does not get proper food or is very sick when growing, that person will probably not grow tall. 2. Changes in temperature directly affect the coloring of the fur of a Himalayan rabbit. Cold weather intensifies their markings, while hot weather causes them to fade. 3. The flowers of some hydrangea plants are blue if the soil is acidic, and pink if the soil is basic. Section F. Many scientists have contributed to the understanding of heredity. The first important investigations into heredity were done in the mid-1800s by an Australian monk named Gregor Mendel. He grew pea plants and collected data over many generations. He recognized that each plant had 2 factors for each trait, one from each parent. Many years later, Mendel's factors were determined to be the genes located on chromosomes. Mendel's work formed the basis for current scientific understanding about heredity. For this reason, he is regarded as the father of genetics. Rosalind Franklin made her major contributions to the discovery of the structure of DNA. Her work with X-ray crystallography provided Watson and Crick with the essential data they needed to visualize its structure. 1951. James Watson and Francis Crick built upon the findings of Franklin and others to discover the structure of the DNA molecule. Watson and Crick were awarded the Nobel Prize in 1962 for constructing their double helix model of DNA. The purpose of genome projects is to determine the complete DNA sequence for each of the chromosomes in the cells of an organism. The Human Genome Project began in the United States in 1990. The first human genome mapping was completed in 2001. Scientists in several countries have contributed to this project. Their goal is to learn what the different genes do and how they affect human health. Section G. Genetic engineering is one of the current frontiers in scientific research. Genetic engineering means taking a gene for a particular trait, usually a protein, from one organism and inserting it into the DNA of another organism. The cells of the organism that receive the new gene will then produce the desired trait. Genetic engineering is used for human health and clinical work. It's also used to produce crops that are resistant to disease and pests, and to improve nutrition and crop yields. Questions about the safety and ethics of genetic engineering exist. Some people question if the DNA of organisms should be changed and wonder what effect the changed organisms will have on the environment. This ends the heredity and genetics section.